Hi, thanks for staying for the last talk, although you're probably waiting for the keynote. Um, so today I'm going to tell you a story about how you can exploit fundamental physical properties of the genome that relate its 1D structure to its 3D structure in order to solve a number of outstanding problems in the field of genome assembly. So ever since the landmark sequencing of the human genome, we've seen all these genome papers coming by, and this has been coupled with the amazing advancement that we all know of high-throughput sequencing technology. And by now, if you're not in the genome assembly field, then you might think that sequencing and assembling a genome with these new technologies is very easy, right? We can get a lot of reads for very cheap. Okay, so before we answer this, let's go very quickly through how you actually do that. So how do you assemble a genome de-novo from short reads? So you have two steps. The first one is called Contig assembly. So you start with a very large genome, let's say 3 billion base pairs. Now you cut it into small pieces and you sequence them with short reads. Okay, and then the idea is very simple. You look for unique overlaps and you stitch them together. Okay, and by the end of this process, now you're going to have, instead of the whole genome sequence, you're going to have just longer pieces, which are called contigs. And the reason you're just going to have a bunch of pieces is because the genome is very large and it's very complex. Okay, so usually this is followed by something called scaffolding. So again, you start with this genome. Now you break it into larger pieces where you approximately know their size, and usually the largest size would be something on this scale, let's say 10 kb, it might be 20 kb or 40 kb, something like that. And now what you do with the short reads is you do paired end sequencing, and then you hope that one end will uniquely map to one contig and the other to another contig, and then you can estimate the distance between them, okay? So you join them together into these larger pieces called scaffolds that have gaps in them. And ideally, by the end of this process, you want to have one scaffold per chromosome, right? So now the question is whether this actually works. So actually it doesn't. And this was very surprising to me when I started working on this problem because I'm not from this field. And Gene Myers actually mentioned this problem. So it turns out if you look at genome papers which are published right now, they use this technique, and the end product is a set of usually 100,000 very small scaffolds. Okay, so they're not arranged relative to each other, it's just a bunch of pieces. And this is pretty bad because actually you don't have really an incentive to go any better than that. Okay, you can get the high profile publication and everything, and doing anything beyond that is quite difficult in terms of high throughput. Okay, so we really have a problem that we can't get high quality genomes like the human genome and so on anymore. Okay, and another problem is that technology isn't going to save us, at least not the current technology. So having more reads or more coverage doesn't help you at all when something is non-unique. And so the current solution to this is basically only low throughput or high cost, things like the old school backs and so on. So basically no one does it. And the strategy currently is just waiting for longer reads to come along, and maybe they'll solve the problem. So we decided to take a different approach to this. So in my lab, we actually work on something completely different. I'm from the Decker lab, and there we developed a technology called Hi-C. So this is a molecular biology experiment, high throughput, and it's aimed at measuring DNA interaction frequencies genome-wide. So the way it works is that you cross-link DNA, and this chemically attaches loci which are close to each other in 3D. Then you cut it with a restriction enzyme, dilute it, ligate, and finally you purify the DNA, and you end up with these pieces where one part of each fragment is going to come from one locus and the other from another locus. So each time you're going to read this, now you're going to read this with paired end sequencing, and you're going to get evidence of a single interaction. Okay, so now you can sequence this with high-throughput sequencing, and you're going to get interaction frequencies, basically. Okay, so one way to interpret it is to look at this map, and so you get this map for the whole genome. Here it's just for 3 chromosomes. So you look at all the 3 chromosomes versus themselves, and now each point here will tell you how frequently 2 loci interacted. Okay? And usually what we do is we try to understand things about the structure and the function of the genome from this. Okay, so how does this relate to genome assembly? So it turns out that there are canonical Hi-C patterns. So in all the Hi-C experiments that we do, in all species, in all cell types, we see 2 patterns which are constant. Okay? And they're actually very annoying, because when you're interested in the biological signal, you're interested in the differences between the cell types or between the species and so on. So what are these signals? One is essentially, now if we look at the whole genome, we see these blocks. We also saw it in the previous slide. Essentially, you see that every chromosome tends to interact much more frequently with itself than with other chromosomes. Okay? And this is probably due to a well-known phenomenon called chromosome territories, in which even in interphase you see that chromosomes are in their own volume in the nucleus. Okay? The second thing we see is a distance-dependent decay. And essentially it means that as loci are further away in the 1D sequence, they tend to interact less frequently. And this is also very well understood. It's basic polymer physics. Essentially, if something is very close in 1D, it's going to interact just by random more frequently. Okay? So we always see these patterns. And now the idea is that this gives us some link between 1D and 3D. Okay? So we have this approach we call DNA triangulation, and the idea now is to measure Hi-C, map it to the set of contigs, and now we have interaction frequencies between these contigs. And now we can use our knowledge of how interaction frequencies relate to 1D distances in order to resolve this into a scaffold. So this is the basic idea. So how does the setup look like? We use Hi-C data. Actually, this wasn't even generated by our lab. This is from Bing Ren's lab. So we use human ES cells, and here we studied 2 problems. The first one is a bit easier. It's called genome augmentation. Here, most of the genome is assembled, and there are a number of contigs where you don't know where they go. Okay, and this is actually the status of all the high-quality genomes. So even the human genome is in this status currently. And I'll talk about that a bit later. And then, given a contig, we want to be able to predict its chromosome first, and then its locus within the chromosome. The second problem is more challenging. There we don't know anything. De-novo scaffolding, we get a bunch of contigs. We want to group them into chromosomes, and then lay out every chromosome. Okay, so let's start with the genome augmentation. So we simulated the following way. We take the human genome, partition it into 100 kb virtual contigs or scaffolds. I'll call them contigs. Then we remove one of them, and we look at its interaction frequency. Okay, so we have interaction frequencies of this versus everything else in the genome. And to make it interesting, we're going to leave out data in 1 megabase gaps around it. Okay, so these are huge gaps. Usually with the current technology, this is much larger than you can bridge. Okay. In addition, most of the Hi-C signal, as you saw previously, is going to be here. So we're also going to throw out the highest and strongest signal. Okay. So now we have this contig. You want to predict its chromosome, and you're going to see that it's just by looking at this, you see it's very, very, very easy. Okay. So essentially, you're getting a row here in the matrix. You need to say which chromosome it came from, right? So we try to use a simple tool as possible because it's got kind of a new type of data. So here we use just a naive-based classifier, and if you just look at the average interaction with each one of the chromosomes, you can build a classifier and you're going to get very good results. So what you see here is the result for the chromosome prediction. These are actual chromosomes versus predicted chromosomes. You have 30,000 contigs, and the contigs are lines, but you don't see them because they're all just stacked here. Okay, and the accuracy is very high, it's 0.999. So this works well. Now we want to predict the locus within the chromosome, and this is also going to be very easy. Okay, so we're going to basically use this signal, and so when you look at the interaction frequency of a contig, this, for example, this contig here, this is its interaction profile. You see it's very, very easy to find where it is. So I'm not going to even tell you how we did it. I'm going to tell you that later, but you see whatever method you can find, you can fit this curve and basically guess that it's here. So it's going to be also a very easy computational task, and these are the results again. So this is for a single chromosome. These actual positions, predicted positions, and you see you want it to be on the diagonal. You see it's very, very good. Okay? So this is already very useful because, as I said, the human genome, for example, is in this state. Okay, so it turns out that another thing that I didn't know, 13 megabase of euchromatic sequences are still unassembled in the human genome. So they have genes there and so on. So usually if you just download the standard assembly, you're going to miss this stuff. And of course, these contigs are very hard to place because until now, no one was able to place them. And we were able to predict the position of almost half of the sequence. And we also had the external fish measurements on some of this, and it matched pretty nicely. So this is already something very useful we can do to given some genome draft, we can now help finish it. Okay, so the next problem is de-novo scaffolding, and this is more difficult for two reasons. First of all, we can't fit parameters here. So if previously we had part of the genome and we could look, for example, how the decay function looked like and fit it, here we can't do that. So we basically need to assume some decay function. Second of all, this is also more demanding computationally. Because if previously, if you think of the number of possible orderings of the contigs, if you want to evaluate just one, then the number of orderings is linear, and here it's going to be exponential. Okay, so most of the formulations here are going to be NP-hard. So the way we simulated this is very similar. Again, 100 kb virtual contigs. This time we leave 900 kb gaps, so we leave every 10th contig basically. So I have a highly fragmented genome. Again, with current technology, you wouldn't be able to do anything at this point. And we try to first group them into chromosomes and then lay out the specific chromosomes. Okay, so to group into chromosomes, we call this karyotyping. Our strategy was very simple. So we take the matrix of interaction frequencies, we convert these into approximate distances, and then we just use hierarchical clustering. And from that, we want to determine the number of chromosomes and then see if the actual clusters match chromosomes. Okay? So I won't be able to tell you, now you can ask me in the questions if you want, how we actually did this, but you can look at intrinsic properties of the clustering. This is the clustering process as it progresses. And if you look at some property, then you can actually find de novo the number of chromosomes. And you see that they actually match the clusters match the chromosomes very well. So again, it's very high accuracy here. So that also works well, and it's very useful. Finally, the most challenging problem computationally, I think, is given a set of contigs that you know belong to the same chromosome, you want to find their positions along the chromosome. So here we used a probabilistic model, and the idea was that we have to make a model, a generative model that explains, essentially models what we expect the data to look like, and then use a maximum likelihood approach to try to find the positions of the contigs that would reconstruct what we expect. Okay, and this here I'll go through the details because it's a bit more interesting, I think. So the model has essentially two components. First of all, you want to model the fact that you're doing sampling here, okay? Because this is very important, because at far distances where interaction frequency is very low, you're going to get a lot of noise and you're not going to sample it very well. So This kind of takes that into consideration. Essentially, this is just a multinomial distribution, so that's like randomly throwing balls into bins, where the bins are now going to be the matrix cells. Okay, so this is why it's kind of looks kind of weird. So it's like the multinomial distribution, except these are now the observed reads, but now it's organized into a matrix. So you have two indices. Okay. Now this is in itself, this is a very bad model. Okay, because now you have a parameter for every bin in the matrix. So this is a huge number of parameters, and we didn't incorporate the fact that we expect to see a decay. So the way we do this is we constrain these parameters by a different set of parameters, which is just this. So we define the probability of interaction between contig at position Si and Sj just as an exponential decay based on their distance. Okay, so you have one decay parameter for the whole genome, and you have the contig positions, and then you have to just divide it by some number to make it into a probability. Okay, so you essentially you plug these two together, and then you get this ugly kind of thing. But it turns out it's really simple, right? Because the x are just the observations that you measure. These are just the counts, and then the only parameters you have here are the positions and one decay parameter for the whole genome. And now you can do a bunch of stuff using maximum likelihood. You can estimate the parameters. So you're going to do the augmentation, which is estimating essentially just one or a few contigs, or you can estimate the position of all the parameters, and that's all the contigs. That will do the De-novo scaffold. Okay? So how do the results look? Again, they look pretty promising. So this is, again, for the simulation I talked about. So you want everything to lay on a diagonal. Again, these are the real positions. This is a predicted position for a given chromosome. And you see it looks pretty nice. Finally, another thing we try to do is work with maybe more realistic size of contigs, because contigs can a lot of times be smaller than 100 kb. And we wanted to work with real data. So we took an actual set that was a set of contigs that was assembled by all-pass assembler in the gauge evaluation. So this is a set of contigs assembled essentially from real data. Here the contigs are now smaller. The median size is 20 kb. And importantly, we don't use any long insert data. So in a real case, you would also have another data set that you could integrate, so your results would be even better. Yet even without that, we can get a pretty good chromosomal layout for the whole chromosome. So you have some errors here, and as I said, when you're going to integrate the additional data set, it's probably going to be better. But again, this is kind of the end point of current technology, so I think it's pretty nice that we can lay out the whole chromosome. Okay, so to summarize, I told you about this method or set of methods called DNA triangulation, in which for the first time you can use short reads to do chromosome-scale scaffolding. It's based on a very simple experiment called Hi-C. It's really easy to do. They do it in a lot of labs. Has high resolution accuracy. It's independent from the long insert data, so that can be integrated. Applicable to any species. Can bridge any gap. We can show that it can bridge even 10 megabase gaps. And importantly, if previously reads didn't help us having more reads, here more reads are going to give us more resolution. Okay. So the final thing I want to mention is just the future, because I think this is kind of introduced a new data type to this field, and I think it was important to talk here because there are a lot of interesting computational challenges here. So there are not only better algorithms given this formulation, but you can also think of other formulations and strategies to solve this problem given this data. You can think of how to integrate this with other data types more integrally. I think there are a lot of interesting things to do here. Also, there are opportunities here, I think, much better than previously. Now you can evaluate how good an assembly is by looking how well it matches what you would expect. There are also experimental challenges. You can also ask me about it, but it's very interesting because now we're trying to think of experimental ways to get rid of the biological data. Okay, usually we try to get rid of what we're using here. So it's quite interesting to think about. And there have been a number of developments since this was published. This was published at the end of 2013. So people have used this same idea to do haplotype phasing. So how do you do haplotype phasing? It turns out the chromosomes, when you have two copies, homologous chromosomes, they're also separated. So if you do Hi-C, then a Hi-C read is much more likely, if it hits two SNPs, they're much more likely to be on the same chromosome. So you can work with this and actually haplotype chromosomes quite nicely. Another idea was to use this same approach to do microbiome studies. So when you do microbiome studies, you take a sample and you have hundreds of different species there, but you can't assemble them into full genomes. So you just get a set of contigs. Now using this, since they're all separated into different nuclei, it's very easy to associate which come from the same genome. Okay? And there really, if you think about it a bit more, there are really a lot of other applications that you can do with this technology. So I really think it's a call for all you computational people to develop this, and I think it's more of a proof of concept and a first step right now. So with that, I want to thank my advisor Job Decker and my group members. This is the paper, and I also want to thank my funding, especially DOE, for supporting my travel to here. Thank you.
Thanks for the very interesting talk. So I'm not sure whether I understand completely. So is the Hi-C data depends on the cell type? So if yes, then the results may be different from different cell lines, right?
Yeah, good question. Okay. So essentially the question was, what about the biological data? Doesn't it actually make a difference? So initially you would think that that might be a problem. That's why you actually have to test it. But it turns out that the other signals are much stronger than the biological data. Essentially the reason is that for each contig here, you have to realize that you have data with all other contigs in the genome. So even if it has local biological interaction with something, then all the signal, the interaction frequency signal with the rest of the contigs is going to compensate for that. Okay, so if I show you even, we have this profile that looked this way. So biological interaction would be like a small peak here or something like that. Okay, so it wouldn't be a problem. But having said that, potentially it could limit the resolution. And as I said, there are ways that we're now thinking of how to get rid of the biological signal. For example, you can, if you're working on a certain genome, you can now pool data types from different cell types, and that would smooth some of the biological signal out. Okay.
Thanks for a good talk. I have a question about when you're doing the sequencing of the Hi-C data set, what sort of coverage do you need? Can you get decent results with one fold coverage, or do you have to go up to higher coverages to get good results?
Mm-hmm. Yeah, good question. So actually the coverage, so first of all, you have to consider this is a scaffolding method, so you have to start with contigs, okay? So the coverage that you need is, I think it's approximately 1/10 of what you need for the contigs. So it's not, or even maybe 5% of what you need for the contigs. So it's not significant. Of course, if you have more, you're going to get better results.
So hi, this is a very clever idea. I was wondering if you have plans to commercialize the idea so that someone could go out and buy a kit and be able to run this in their own lab without having to work out all the details.
It's complicated. I hope so. There's some issues with IP and so on. We don't have a patent on it currently, so this is also being used by. So this was published back to back with a group that essentially did something very similar, and with a haplotype phasing paper. Um, I don't know at this point.
Yeah, it'd be useful though, because I mean, I could see labs taking advantage of this, you know, throughout the field. So it'd be good if some way you could find some way to get this out there so that people could
Sure.
just buy it and do it.
Yeah, thanks.
Sure.
So the resolution of Hi-C is kind of okay. There's an intrinsic resolution, and that's the kind of the theoretical limit is the restriction fragments that you get, and that's about 4 kb with the current enzyme that we use. That's really the limit on the resolution. Beyond that, the resolution that you're talking about really depends on how you bin it. So it's true that if you look at smaller segments, you'd have less data. Currently, I don't think it's a big problem, but you're right in the sense that for very small contigs, this won't work. Right. Right. So I kind of show you that it can work for like maybe 20 kb, 10 kb, but for very small contigs, it probably won't work.